Welcome to the Laboratory of Molecular Microbiology. We are at the Department of Food, Environmental and Agricultural Sciences at University of Foggia. Our group is involved in a project funded by the European Union in the 7th framework program called QUAFETI. Fresh cut, packaged, fruit and vegetable sold in the market are considered a product of high quality and freshness. However, they may represent a risk due to possible presence of pathogenic bacteria that can cause serious illness to humans, like Listeria monocytogenes and E. coli O157H7. From an industrial viewpoint, the prevalence of these foodborne pathogenic bacteria is a serious threat for fresh-cut producers because it determines a significant economic loss due to the loss of the products, to the withdrawal of batches of contaminated foods from the market, to the damage of the company's image impacting its intangible capital. The existing regulations in Europe provides that the identification of Listeria monocytogenes and Escherichia coli O157H7 in foods is carried out by culture-dependent methods. Quantification of Listeria monocytogenes and E. coli O157H7 in foods is generally done by the most probable number method, which requires complex and time-consuming analysis. For example, the detection of Listeria monocytogenes requires successive steps: a primary enrichment in a selective broth medium during 48 hours, ability to grow on selective plates agar after 24-48 hours, confirmation of the presumptive colonies by biochemical assays, another 24 hours of time required. Now we know that fresh-cut products have a shelf life of 7 to 15 days, and the official diagnostic methods may take up to 12 to 14 days to detect pathogens. Thus, for companies, it is very important to have a faster and reliable detection methods. In the last years, a considerable number of detection methods using faster molecular tools have been developed. These techniques involve the extraction of microbial DNA from food matrices to use as target for a selective amplification reaction called polymerase chain reaction. Polymerase chain reaction is a molecular approach based on the amplification of a target fragment of DNA, in our case, specific for the microbial species that you want to identify. In the real-time PCR, this species-specific DNA fragment is labeled with a fluorescent probe that allows to monitor in real time the amplification process. Thus, it is possible not only to determine the presence of a microorganism, but also to quantify its initial concentration. Based on our investigation, we propose a most probable number coupled to real-time PCR method. For example, we are able to quantify Listeria monocytogenes after only 6 hours of enrichment in a selective broth. At the same time, we are also able to detect Escherichia coli O157H7. Furthermore, we proposed to use real-time PCR to always confirm the positive sample to most probable number method. This operation is reliable and requires only few hours of time after an enrichment step of 48 hours in a selective medium. Our protocol works. A portion of your product is weighted and then placed in a sterile bag that contains the selective enrichment media. The sample is homogenated in a blender. Homogenate is then diluted 3 to 5 times and placed in tubes for most probable number. After 6 hours of incubation at 37 degrees Celsius, we pick 1.8 milliliters of the homogenate and go for DNA extraction. DNA is used for real-time quantitative PCR. It takes about 1.5 hours. At the same time, the conventional most probable number method goes on. Then we have black tubes, which are presumptive positive, and yellow tubes, which are negative. By classic method, you need other 3-4 days to confirm the positives. By real-time PCR, you need 3-4 hours. Results are 100% confirmed. What are the drawbacks of these methods? Equipment cost and skilled labor. Samples should be delivered to a biology lab equipped for PCR. But there are also many advantages for the companies. Since this method is fast, reliable, and allows a earlier detection of pathogenic microorganisms, even if the official methods have to be performed for final confirmation. Nonetheless, the proposed method allows you to intervene promptly on presumptive contaminated products, to properly sanitize the plant, a better control of the quality of your product, and to ensure greater safety to the consumer. Prevention is better than cure.